I think I'll take that as a cue that we're good to go. So hi, welcome everyone. My name is Dluzen. I'm going to be moderator and kind of panelist today. We're missing a few people today, but that's okay. We're still going to have a great conversation. They might be joining later, so life happens, and we're gonna have a great conversation. So I'll introduce the other panelists in a second. So again, this is our science track, CRISPR for spacers, and we have a kind of a prompt today. So, the prompt for this discussion is that we're imagining we're, 2121 and we're aboard the generation ship Nostromo. And what gene mods would we need to travel into and to colonize other worlds? Perhaps not just for ourselves, but for animals, plants, and other microbial co-voyagers. What would you need to be able to do once you got to the new world? So today our discussion is going to be about genetic technology and how we would use that to slip the bonds of Earth and integrate into and survive extraterrestrial ecosystems and work with or defend against alien life forms. I will introduce Sandy in just a second. We're going to heavily rely on the fact that you all have wonderful burning questions to contribute to our discussion, so I'll be monitoring that. A little bit about me. I am my name is Dluzen. I'm a human geneticist. I study aging-related diseases and health disparities in human populations. And I write both nonfiction and fiction that focuses on gene editing and the societal ramifications of gene editing. And I'll turn it over to Dr. Sandy Gibbons, and you can maybe perhaps give a little background about yourself.
Absolutely. Thanks, Doug, and thanks for inviting me to be on the panel. This is really cool topic. So I am a research microbiologist. I'm at the U.S. Army Combat Capabilities Development Command, otherwise known as DevCom, Chemical Biological Center. And we do typically do a lot of work with synthetic biology and in terms of usually biological defense, but recently, we're expanding more into other areas of synthetic biology, such as biomanufacturing. And I've recently I've been asked to serve as technical lead on the scale-up facility for biomanufacturing that we're doing down there. And so this is the discipline of taking getting microbes to make stuff for us. And it's been something we've been doing for 100 years now. Overall, but with the new genetic modification and engineering technologies really become exciting. And so I think it's a good time to try to talk about some of these panels. So that's it.
Absolutely. Awesome. So I think what the kind of the shape of the scope I'm thinking of for today is maybe you and I will just have a few minutes discussion a little bit about gene editing and its role in society, and that we already have a couple questions, which I see in the quick Q&A, which is awesome. So thank you. Well, I suppose we're going to be talking a little bit about editing microbes, maybe other organisms, and perhaps ourselves. And so I think one thing that we should discuss perhaps is, what are your thoughts about editing the genes of humans and maybe the different types of editing that there is, such as germline versus, adult cell editing, and we'll probably have to discuss a little bit about the ethics of that, and then maybe set that aside as we're kind of speculating what we would just need to do if we were going to go on a transgenerational voyage across the Milky Way.
Yeah, that's obviously, it's an area that's fraught with ethical complications. And, the science fiction is filled with stories of things gone horribly wrong and things gone right. What's been on my mind, of course, lately is and here's my cat. My daughter. But anyway, and so this is really the place and the venue in which to deal with some of these ethical, legal, social, and types of questions, because this is where ultimately we get to speculate without restraint. And so, so there are two different, to talk about gene editing of humans, we've gene edited pretty much everything else at both somatic, which is sort of when you're talking about cells that don't transmit into the next generation, that would be somatic gene editing. So if you're editing cells in the pancreas, say, or if you're using a gene editing to neutralize a cancer-causing modification or cause cancer cells to express something that will get them killed by the immune system. Sit down, please. Thank you. You can listen, but please. Anyway, but in terms of germline modification, that is where you're creating a mutation that is transmissible to the next generation. And where the ethical questions on somatic mutations are significant enough as it is, once you start visiting the sins of the fathers and mothers, so to speak, onto the 7 and ultimately potentially infinite number of generations going down, then the ethics become even more severe. Now, that calculus may change when we're talking about transgenerator, getting off of this rock and going somewhere else, because your needs are changed. The costs may change, the benefits, the cost-benefit analysis may change. And so while we may decide to leave those of us here stuck on planet Earth alone, societies may decide to go ahead and allow this sort of thing to allow survival off-world and into subsequent worlds. That said, also, mutations that may be advantageous for survival in space may not be advantageous once you get down onto LV-426 or something like that. That's been on my mind lately for, if you want to come to my panel on invasive species tomorrow, that's a shameless plug for that. But
it's all good.
But anyway, the calculus may change. And the thing about gene editing these days is it is getting easier to do. It is getting more portable. And the infrastructure required to do that can actually, and I know that NASA is working on being able to do this in space and on the fly. And so what gene synthesis now can do, because all of this requires the ability to synthesize DNA from scratch and create the mutation that you want, you may be able to then reverse it later. And I saw a question in the chat about what happens when you make a mutation and potential off-target effects or unpredicted, law of unintended consequences. Yes, that is a huge question, and that is why some of these editing experiments in humans have been said or have been outright nixed.
You're right, and to put it in perspective, as we leave Earth, currently there are several clinical trials going on around the world that are using gene editing to cure diseases in adults. Cancer and cystic fibrosis are two of the main ones at the moment, that these are mutations to, you mentioned, our somatic cells, cannot be passed on to progeny, cannot be inherited to your children. But there was 2 years ago, kind of a major shock throughout the scientific community in that a researcher in China performed CRISPR editing on a pair of twin babies in the uterus. And there are now, at least as far as we all know, at least 2 children that have been born that are the first ever to be modified by CRISPR and edited, that will also have that modification in their germline. And so if they choose or decide to have children in the future, they would be passing on this modification. And the edit was to alter a gene that would protect the children from HIV because the father of the children had HIV. That current researcher broke several international laws and guidelines to perform this work and has been stripped of their medical license and jailed for it. And there have been a lot of international discussions on how far should that technology be pushed as it currently is today. And so, as mentioned, the question of the off-target effects, it's really unknown. This is a gene that's involved with the immune system that allows HIV access into the cells, but that gene may be involved in other processes. And so the downstream impacts are going to be unknown until those children get older. So that kind of just frames where we are currently with the technology. I saw there's several questions in there. So Sandy, I'm going to throw one that we have here in the question and A to get our conversation going about what happens when we get out into space. How much infrastructure would it take to have a CRISPRing society? And say, could the planet and aliens keep CRISPRing, assuming non-hereditary CRISPRing?
So I can answer some of the technical infrastructure aspects of that. You need a laboratory, first of all. And those are getting smaller and smaller and smaller. 10 years ago when I started at CBC up here in Edgewood, Maryland, the sequencers, the things that read the code, the instruments that read the code were easily on the bench top, if not larger. So you think of a decent sized coffee table, they would occupy that space, and then you would have the entire laboratory infrastructure to support that. Well, last year, some of my colleagues went out and into the field, with a bunch of soldiers and did sequencing operations out there, identified an organism using a sequencer that was no bigger than a thumb drive. And in fact, it plugs into a USB port, which gives our InfoSec people some heartburn. But that's the kind of miniaturization we're talking about with sequencing technology. Synthesis technology is experiencing a similar downsizing and miniaturization. And as microfluidics develop, that's going to be even more prominent in that. So that I'm going to go out on a limb and say that the laboratory infrastructure that you're going to need in 20 years or 30 years will easily fit into the space of a desktop computer.
Yeah, there are definitely
reagents and the supply chain for the reagents. That's another story. But I have some thoughts on that too down the line.
That's a great point. The enzymes needed, and there's a new type of sequencing technology called nanopore sequencing, and that's what I was referring to. Yep. Yeah, those sequencers, they can fit in the palm of your hand, there's some fidelity issues with the ability to get good reads from them. But I've
sequenced a microbe to precision. I mean, it was awesome. Good enough.
Awesome.
And it was a bug I was doing some gene editing on. So, and inserting genes into the genome. And we were able to see that beautifully, just with nanopore sequencing. So the technology's there. But so, the real issue, I think, is going to be the synthesis. And that's probably lagging about 10 years behind the sequencing technology. So you write the code, you read the code, you write the code, and then there's the matter of interpreting the code.
Right. So going back to the Nostromo, we have our laboratory, we have our sequencing. We're assuming we have the reagents to perform the sequencing. We're assuming we have the reagents to create the mutation, whether it's via one gene editing tool or another. CRISPR, of course, the big one right now, but perhaps even a better one is discovered in the future. Let's start with kind of in your world, Sandy. Let's talk about. We're getting a lot of great questions, so Russell, your question about synthesizing new organisms from scratch. I'll take that one real quick. So, how far away do you think we are from synthesizing new organisms from scratch? Within a decade, there are several organisms, small, like yeast and smaller, that there are now full synthetic chromosomes for. So the yeast synthetic project, I believe there are probably, and you'll probably know more about this than I will, Sandy, there are several bacteria that I believe the viruses that have been completely synthesized. And that's really taking the genome, say, from a bacteria, and you can build it end to end synthetically, but you can also then manipulate it and change it and rearrange the genes and invert the chromosome order if you want. So that's a whole field of synthetic biology that's really took off in the last 5 or 6 years. And the plan for the yeast, a full yeast artificial genome, is within the decade. So humans would be much more complicated.
Well, yet, yeast has, I think it was 8 chromosomes, I think, or something like that. They're small in this compact genome. The trick about the human chromosome is that it has a lot of features in it that make it difficult to sequence and make it difficult to synthesize. So the idea that you're going to synthesize a human genome is still very far-fetched.
Yes.
But to tweak it, you need maybe, You don't even need to have the enzymes in place. If you can synthesize the DNA and assemble it in a yeast cell, for example, you can get that plasmid and you can dump it in, and you can then put it in your tissue culture media,
right.
In your tissue culture, and you're off to the races potentially.
So Jacob asks a really great question. I think this will be a good framework for moving into some of the things that we would practically need to do. And I think we can frame looking at this from the perspective of human physiology to support systems for us, and then our microbial friends that are coming along for the ride. So Jacob had asked, what are the mods for two key issues in space: radiation damage and microgravity? And I think that we can pull right from here now. What do you see as being the most important things that we're going to need to consider?
Well, radiation is clearly an issue. Ironic, in my talk last year was on how to kill spores with radiation, in part. And so, it is a significant problem. And obviously, as you get higher and finally out into interstellar space, the radiation damage is potentially severe. So you would need to jack up the DNA repair pathways, primarily potentially for the non-homologous or for homologous end joint or non-homologous end joining pathways, which link broken strands of DNA. Because unlike the DNA damage that you're getting from UV light that causes, say, melanomas and things like that, that's all DNA damage. Or from certain chemicals that intercalate, between the base and ladder, PAHs and things like that from your grilled chicken. Unlike those, what you're getting in outer space is high intensity ionizing radiation. You're getting gamma radiation that's highly penetrating, and you're getting cosmic rays, the helium nuclei that is flying, alpha particles flying at ungodly amounts of energy. So those, in contrast to the base modifications that UV causes, those cause double strand breaks. And those are generally lethal events, which is why CRISPR actually works, because CRISPR, like other recombinating techniques, breaks the DNA. And then the only thing that lives is the stuff that's actually fixed the break. And you provide the template by which that break gets fixed. So you need to find a way to jack up or increase both the frequency and the fidelity of DNA repair, strand repair. So that's one. Microgravity is a whole nother question. And I honestly don't have a full answer to that because I don't have a great understanding of the physiological effects.
So microgravity is really interesting. One of the famous study. Oh, yay! Emily's joined. Hello,
hi, Emily.
So our other panelist has joined. Emily, I'm just going to finish answering this question, and then we'll introduce you too. We could have a 2-day discussion on the things that happen to the human body when you're in microgravity. There's a really great study. So I'm going to type into the chat box there, called the NASA Twin Studies, which took 2 astronauts that were twin brothers, Mike Kelly and his brother. What is his brother's name? Scott Kelly. In fact, one of them is now a senator from Arizona. And won in the election in 2020. They are identical twin brothers that have gone through similar training at NASA, and they did an experiment where I forget which brother went up into space for a year and then came back down. Thank you. That's the great NASA link there. And they studied the genetics, the physiology, the microbiome, the bone structure, mental health issues of the astronaut in space versus the ground control. And this was a really interesting study, even though it was only 2 people. Genetically, their background is extremely identical. So that allows you to control for a lot of things that maybe the genetics would have a say in. And you're really then looking at just environment. And what they found is that for the up in space, certainly there is more DNA damage. There are issues with your eyes and not cataracts, but developing macular degeneration, that the muscle around the eyes weakens. There is your bones will elongate, not your bones, but your limbs will elongate, and you'll get a little bit taller because the pressure of gravity has alleviated that process a little bit. And interestingly enough, tying back to DNA repair, our telomeres, which is the caps of our chromosomes, will actually expand a little bit. And what we found is that when we age, as our cells divide, we lose a little bit of the ends of our chromosomes. And that actually rate limits a little bit of how many times our cells can divide. And what they found is that in space, counterintuitively, those telomeres expand a little bit. And that might be a way of promoting longevity and healthy cellular growth on a transgenerational ship, is not only protecting DNA damage and DNA repair, but also extending the lifespan of our cells by, inserting in telomerase, or another enzyme that actually maintains the functionality of our chromosomes.
You got to be careful what you wish for, because when the telomerase reactivates and other things like that, you get cells cancer. That live forever, and we call that cancer.
Yes, absolutely. So there's a fine balance there. And the same with DNA repair. DNA mutation is a natural part of how variation is created in the human species. And so if we are to kind of stop that process, every time in a generation ship, for example, someone has children, they might become genetically identical to one another. And then we have a problem of not enough diversity in the gene pool as we are traveling across the stars.
Not to mention meiosis, the crossing over events are recombination events. And if you stop that pathway, you've nixed your genetic diversity. Right. Anyway. So I'll stop talking now.
No, that's a great point. So there's a lot to consider with those kind of unintended consequences. Emily, I would like you to introduce yourself and a little bit about you. We're just in the process of talking about how we're going to get to another planet and what are the types of things that we might need to edit along the way. So welcome.
Thank you very much. I apologize deeply for being late.
No
weekend
problem.
Stuff at home. Anyway, I'm Emily Lakdawalla. I'm a science writer, a planetary scientist. I studied geology on Mars and Venus, but I've spent most of my career writing about the exploration of space by semi-autonomous robots, the rovers exploring Mars, the orbiters exploring Saturn and Venus and everything else. I've written a book on the design and engineering of the Curiosity rover, and I'm writing a second one on the science from the mission. So I'm rooted in deeply practical exploration of the solar system by robust machines that can go places that humans currently can't. And so I'm glad there's some experts here to talk about the biology. I can talk about the machines.
And potentially about the difficulties of actually getting, why we have to send machines in some of these things. You know, why humans just don't do well in an environment like that.
Yeah, absolutely.
Yeah.
So we have a great comment, I think that's going to lead into both of the expertise that you both have, is that, so Jack Curl asks, an alternative to the premise is that the spaceship travels without any biological beings on board to avoid radiation damage and the ethical problems of confining generations to the ship. But then after the arrival, the original problem arises of when it's time to build a biome from a clean sheet of paper to fit the new environment, including building humans to live there. What do we need to consider?
Diversity, diversity, diversity. If you look, for example, at the microbiome of hunter-gatherer societies versus our microbiome, you find a hemorrhaging of biological diversity in the microbes that live in our gut. And so what we're dealing with today is much less diverse than what we were dealing with, hundreds of years ago. We also have to deal with the microbiomes for all of our pet animals that we bring along, Doug, your cat, but any livestock we want to bring along, and on all of this is an informatics nightmare, because the amount of data that this requires is phenomenal. So you're going to have to encode the data in the most information dense material we have, which is probably DNA at this point. So you can use DNA for encoding things other than life. You can encode the Encyclopedia Britannica. You can encode movies, and this has all been done. A lot of it by George Church up at Harvard and places like that. But a lot of this, so DNA is an information storage media. That's one thing. But in terms of, but what I think you really need more than anything else is diversity, because diversity gets you versatility.
Mm-hmm. And Emily, how about infrastructure? What are we looking at if we're going to do something like this?
Well, I'm a big fan of exploring the connections between humans and machines so that you can maximize what you can do in all of these different environments in space. By taking advantage of the robustness of machines, you can send them to places that are higher temperatures, higher and lower pressures, nasty radiation environments, cancer-inducing radiation environments, and all of those things, and keep your very fragile sacks of flesh inside carefully contained environments that are designed in order to keep the humans alive. And so even when we're talking about much nearer to home kinds of concepts, like sending the first humans to Mars, and I'm sorry, I'm going to have to mute for just a second as there's an airplane landing over my head.
Well, while Emily is muted, something else to consider is also when we talk about biodiversity, and Emily, if you can also comment onto this, is that one of the questions that Daniel has is that would we become biome locked? Would we have the fact that we're living with plants and animals that we're exposed to during either the transit process or once we get to this next world? Is there a way, maybe it is inducing new mutation to create that diversity? Would we become locked with those organisms, and is that good or bad?
Well, my point is that you don't necessarily need to adapt the human to the environment so much. The humans, more and more, we're already, especially this last year, we're living in virtual environments. We're doing our commerce, our interaction with other humans, and everything like that from safe enclosed spaces. And so it's not much of a stretch to imagine that when you're talking about exploring other worlds with much more advanced technology, that very soon we're going to be talking about walking around with bodies that aren't our own. They'll either be robots, or you can imagine some other kind of more organic engineering in the future. But you don't actually need to make the environment change so much. And especially if you're talking about looking for life in other worlds, it's actually unethical to change the environment very much before you know what kinds of inhabitants there may be in the place that you're exploring. And so I'm a big fan of not changing the environment to suit the human, but to give the humans different suits to go out and explore the environment.
Yeah, come to my panel tomorrow, and that'll give a sense of how good we have actually been at adhering to the ethics of terraforming. It's a great point. In terms of the bio-lock, the founder effect, if you look at, say, the migration across the Bering Sea, it was a very small number, relatively small number of individuals, or as we, as humans, we traversed out of places like Africa and colonized Europe and Asia. The genetic diversity in human populations in those is much, much, much smaller. And especially in the Americas, where the founder effect is particularly strong. And so that is where we see founder effects, not only for the human population, but for all of the populations we bring along with us. However, with synthesis and CRISPR, that is no longer as big of a topic, because we can then ostensibly, we can then start to artificially introduce the diversity that was lost during transit.
Correct me if I'm wrong, Sandy, but isn't there a pretty big founder effect in nearly all of the human population except within Africa?
Yes, that's true. Yeah, that's absolutely true. And even there, I think at some point there was a geological event like a volcano, and it wiped the human population down to maybe 10,000 individuals. And so to think of the genetic diversity that was lost in that bottleneck event, as long as you retain the information in bits or in DNA, better yet, in DNA that you've stored in a tube, your ability to recreate becomes significantly more robust. And as long as bioinformatically, if you do compression by reference, you can store even more DNA in that, or more information in a limited amount of material.
Mm-hmm. Well, Ed asks in our Q&A a question that I think is perfect along these lines that you had mentioned, bottleneck effect and genetic diversity. And his question is, how many genes would you have to edit in a human germline and still call the progeny human?
It's definitely not a question for me.
Well, I can take a stab at it. You mentioned the bottleneck effect and that reducing genetic diversity, and in evolution, the idea of speciation, of changing a species from one to another, has everything to do with diversity and variation. And as soon as two species have, or say two individuals that have enough variation between their genomes that they cannot produce a viable offspring, and by viable, I mean an offspring that is also capable of producing offspring, then you have two separate species. So again, like an example would be a liger, right? So that's the progeny of a tiger and a lion. You can have adult ligers, but they are sterile creatures. They are incapable of passing on their own genetic information to their next generation. And so at that point, I don't know if there's a magic number in humans, at least, of what you would need to do. I suspect that it would be a considerable amount, given the amount of variation that's in the human genome already. Everyone's genome, so everyone in this panel, everyone in the audience right now, we all have the same human genome, same kind of build of the genome, but we all have variation in it. Millions and millions of our base pairs of our 3 billion base pair genome are different between any two people. And so it's a considerable amount of variation. We would probably have to really, jack that variation up even more to be then considered a different species, or perhaps a subspecies, when we're talking about how much variation there is. I don't know if that would be different, say, for a microbe. If that would be, probably because of the smaller genome, maybe fewer changes would be needed. But I don't know.
Well, microbes are a whole nother kettle of fish, because they like to swap entire sections of genome between organisms. And so you can, if you look at this is close to my realm, if you look at the difference between Bacillus anthracis, which is anthrax, and foray, which is Bacillus thuringiensis, which is sprayed all over the place on to control gypsy moths around the area. They're not that far apart at the core. In fact, they all kind of nestle within a group that also causes some food poisoning type things. And yet they're not lethal to us, and they're considered different species. Yet are they? Not really. Bacteria's speciation in taxonomy is a mess right now. So let's not even go there. But the idea that you can change something rather subtly, or with a couple of things, and get a different species, nah, I'm not sure. But you're right, in terms of compatibility. One might argue in some ways that the dog, the Chihuahua, and the Great Dane are two different species, because biomechanically that simply might not work so well. I don't know. I've never tried that experiment. I don't think I want to.
Well, Jennifer has a question for us in a Q&A, and I'll add another one on top of it for both of you. So her question is, what about editing our own microbiome instead of us, but our microbiome, our co-inhabitants, as a way to help us adapt to a different environment? And Emily, what are your thoughts too about bringing microbes to help us adapt to the environment? So I know, like, say with the Curiosity rover, the big push is to make sure we're not introducing anything. What are your thoughts about if we would need to bring something with us to help us survive, or are we going to be able to do it all just in our suits? So I'll open it up for you both to comment on that.
Well, obviously, we'll have no choice about bringing a lot of microbiome with us. It's in us all the time. It's what helps us digest food and do a number of other things that I'm sure Sandy would be able to tell you much more about. And it is also true that we're working on various technologies to use microbes to help with environmental change on Earth. There's work to engineer microbes that can, for instance, break down oil that's been spilled into a groundwater environment. And so there's certainly work being done along those lines. And as Sandy mentioned, it would be, you could imagine bringing along the kind of DNA recipe for those sorts of organisms and finding out what you need and maybe tinkering capabilities together. It's already what we do when we're making genetically modified crops, is to insert useful genes into crops that already grow in order to make them better adapted to a different environment, a drier environment, or one that contains certain toxins or whatever. So it's not too hard to imagine doing that. And then there's also going to be the free riders. There's going to be the stuff that we can't avoid bringing along with us, and who knows what'll happen to that as it gets introduced into a new environment. When you're talking about the timescales involved in interstellar travel, that's a lot of time to to allow microbes to evolve and do things. And who knows, you could wind up with a COVID event on a spaceship. That would be pretty bad.
Be awful.
But presumably if you've got faster than light travel or interstellar travel, you've got technology to deal with those sorts of things. The sad truth is that we don't have the technology we need to send humans to a base on the moon yet, much less beyond our solar system. So we got a long way to go, but it is fun to write the science fiction.
Do you think it would be useful to bring maybe a library of microbes? I know extremophiles that live in the vents of undersea volcanoes as something to say, oh man, we're landing on a volcano planet or a hot sulfurous planet like Venus, for instance. Maybe we need to bust out some of these and see if we can get some assistance.
I mean, it depends on what your goals are. So ultimately what you're talking about is something that in the space world we talk about is called In-Situ resource utilization, or ISRU. And the idea is that you bring along the know-how that you need and maybe a couple of tools to make use of what you find in the environment when you're there. So the big one that we talk about for human travel within the solar system is just getting water out of apparently dry environments. Mars and the moon both have water locked in their crystal structures of their minerals. And so there's lots of work being done to figure out how to liberate the water from those materials, because humans need water not only to drink and to wash and all those things, but you also breaking down water into hydrogen and oxygen makes a terrific rocket fuel. And water is also really good at shielding people from radiation, which is very damaging to humans. And so water has all these useful functions, but it's very heavy. It's probably one of the main things that we spend money on launching to the International Space Station right now is more water. And we reclaim it in all the ways that we can. I mean, yesterday's coffee is tomorrow's coffee. So, it's how things work in space. And so the other thing that they're working on for ISRU, In-Situ resource utilization, is building materials. So taking dust on moon dust basically and making bricks out of it, which is one of the first things that you have to do. You have to make shelters against solar storms and stuff like that. So if you're talking about bringing microbes with you, presumably you're talking about similar things where you would be using those microbes to metabolize materials that we can't use into materials that we can use. So perhaps you would have the microbes on a volcanic planet manufacturing oxygen for you, or fuel, like say turning methane into longer chain hydrocarbons that could be stored more safely, things like that. You could imagine all kinds of uses for microbes in that way.
Yeah, and there are active programs and research, some of which I'm now starting to get involved in using microbes to actually, I'm not doing the building materials, but there are actually microbial concrete, for example. Yeah. Microbial concrete, there's microbial building, insulation. You can get there's certain yeast species or fungus will then their mycelia can then be turned into insulation foam, for example. So there are tremendous, and then using it to make any sorts of things from precursors to composite resins to using to melanin for radiation protection and that sort of thing. So there's a whole slew of things that microbes can do for us in terms of industry through fermentation. We've been doing it since we started making acetone for gun cotton in the 1917 timeframe. So it's a huge industry. But now with all the DNA tweaks that we can do and the metabolic engineering we can do, we're learning how to channel metabolites into where we want them to go to make the materials that we think we need. That can then be potentially done on the fly.
Yeah, and Jeremiah asked a follow-up a little bit to the beginning of our discussion, Emily, when we were talking about just like laboratory needs to create new species. And his question is, if it's a one-way trip and an unknown destination, I could see trying to bring a library of species genomes along. What kind of infrastructure would a new colony need to begin turning that library back into useful microbes? And we talked a little bit about being able to sequence and synthesize sequences. I would also add in that we probably just need even conventional 3D printers to be able to print new lab equipment and print proper PPE or proper, maybe even printing our food from the basic raw materials. What are both of your thoughts on other types of things we might need to be able to kind of do that kind of bioengineering?
Yeah, that's a great question. And, ultimately, yeah, you mentioned it, you need synthesis, you need sequencing for QA, you need, and then for a microbe or for any living organism that reproduces from a living organism, you need a template. So as far as right now, we don't really know how to restart something from scratch. We have rebooted an existing organism with a new genome, for example. The first bacterial genome was done that way. But we haven't actually taken a lipid bilayer with a mix of chemicals and injected DNA into it and gotten it to start up. You need some kind of living template organism. Once you have that, then you can perpetuate that and modify it accordingly. But it might take a long time.
I can see you having to work from the ground up, like having almost like a civilization tech tree, where you start with something extremely simple, and that from that you can make X, Y, and Z, and from that you just wind up bootstrapping yourself all the way up the phylogenetic tree.
Mm-hmm. Absolutely. So I'm going to go back to a question that came up. We'll change course a little bit and talk a little bit more about humans again. So someone posed the question, what about preparing the humans for a generation-long journey by enhancing the empathy centers of the brain? And I'm suspecting that this is in the lines of just making us a little bit more collegial with each other, perhaps, or sympathetic with one another on our trips. Any thoughts about ethics? Aside or not aside on
less
some
competitive,
of that.
Perhaps. If you go back, if you look at the classic xenogenesis, Octavia Butler's story, where humans basically are locked down on the planet because we're too competitive, and so that and alien life forms come in and say, oh no, you don't. You're not going anywhere. Yeah,
right.
And so there's that idea and that modifying that, but to some extent, where do we get to the point where we're tweaking and we're not human anymore? We're something else. We've become transhuman in a way.
There's been a lot of discussion in the chat box between the panelists. So that's great.
This may be my practical nature coming in, but I have a feeling that drugs would be a simpler solution than trying to alter the empathy centers in the brain.
And we can get microbes to make those for us.
Exactly. You can do an awful lot with drugs. I don't mean to be flip. You can actually really manipulate how people behave, how they think, how they act. And it seems to me that it's a lot simpler to begin with, to work on drug cocktails that make humans behave in ways that are better for ships that you're locked on for a long time, than to try to actually alter the genetics.
Yeah, there's been some interesting studies too. So in at least in human research, and they're called Genome-Wide association studies. And the idea, for maybe those in the audience that aren't familiar with this, I'll type it in the box. It's pronounced GWAS for short. And that is, you study a large population. Say you study 100,000 people with Alzheimer's. And the idea is by sequencing their genome and knowing that they have Alzheimer's, you might be able to find genetic markers associated with that trait. And so this has been done a lot for a lot of different diseases like cancer, Parkinson's, Alzheimer's, diabetes. And a lot of interesting genetic markers or susceptibility markers and risk factors have been identified, different alleles, different variants, perhaps associated with risk or protectiveness against these conditions. In the last couple of years, researchers have branched into looking at genetic markers, say for intelligence, or for someone mentioned sociopathology or for other types of behavioral traits or environmental traits. And one thing I would like to mention about that is when you put together what is called, and I'll type this into, a polygenic risk score. So analyzing your genome and what does your genome say about your risk for Alzheimer's, or what does your genome say about your IQ? There's a couple things to keep in mind with that. One is even the most robust studies only account for about 5 to 6% of variation in the human genome, particularly in studies on intelligence and on empathy and other social behaviors. A lot of it is learned behavior. A lot of it is much more about your environment than it is your just genetics telling you that you are, a sociopath or you're going to have a high or low IQ. And even the IQ test is something that is debated as whether or not that's even a useful marker of intelligence. So,
and then there's an additional component, if I mind me jumping here, of epigenetics, where you can have covalent modifications to the DNA, but the sequence doesn't change. But now you've stuck little markers on it that say encountered stress here, and that changes patterns of gene expression that can be transmitted to the next generation.
Right.
And you see this in the children of survivors of the Holocaust, for example, where it's been shown that there are some heritable markers that are associated with trauma and stress.
Right.
I would also put a big red flag on any study of IQ or intelligence or emotional stability or anything while we still live in a society that is based on prejudice and racism and sexism and everything else.
Absolutely.
You really can't rely on the results of any of those. And science fiction has to be real careful because of its long history with being associated with these scientism kind of over-reliance on science being kind of objective and true, when in fact it's like everything else we do. It's done by humans who bring their own prejudices into things. So you have to be careful about that. But the other thing I wanted to mention that was kind of in context of this discussion was the fact that the things that the characteristics that make humans able to live on generation ships for a very long time are not going to do them very well when they suddenly have to be very entrepreneurial on a new planet where they arrive. So one could imagine having gen ships full of people who are basically drugged for generations, and then suddenly the drugs are taken away as they arrive, and you have a new generation that's never experienced that kind of thing before. That would be kind of fascinating.
Absolutely. And to follow up on your point, Emily, about understanding the genetics of what our diversity is, that when you look at the field of genetics, for example, it is riddled with eugenics and a lot of really kind of just dark history that people have tried to use genetic markers as a way of instilling their social prejudices. And so that would be something that would absolutely have to be discussed in a group of people that are going on a transgenerational voyage on how to prevent and making sure that if there were going to be modifications in any gene, is it being done for the right reasons and for what reasons and what those criteria are. And those are discussions that are happening on Earth today about how we even do that in health research. So there's not a lot of solid answers for some of those things yet. I think we're getting real close to our sign-off time. So I'll start with you, Emily, and then to you, Sandy. Just any last-minute follow-up points you wanted to make on this topic? I really appreciate your time today and this awesome discussion.
Yeah, I'll just pitch in one particular idea here, is that we don't necessarily need to be sending a spacecraft in the way we typically think of it. As a structure of metal and plastic. The ideal spacecraft may be something that carries that microbial and plant and whatever diversity in a sort of hybrid structure that is itself alive and self-healing and can confer many of those essential living functions, perhaps powered by a fusion reactor at the core to generate heat and stuff. But is a hybrid structure between living and non-living or inorganic materials. And God knows what that's going to look like by the time it gets there.
And I guess my closing words is, you know, science fiction is often a way for us to escape the problems that we have back here on Earth. And we have to watch out for people who really are trying to make science fiction reality, who are not confronting the problems that we do have to solve here on Earth, lest we export them to space. So I'm thinking about, you know, people who are like, oh, we need Mars as a backup planet. Well, Earth is still the best planet in our solar system for us to live on. And we got to make sure that we don't ruin this home world and think of Mars as a place that we can escape to, because we can't. You know, this is always going to be our home world unless
Absolutely.
We have Daniel and Giskard, you know, lighting up the plutonium reserves in the crust or whatever it was that ended the Isaac Asimov series. But yeah, it's going to be our home world forever. So we gotta treat it well. If
there's a Twitter or a website that either of you have that you'd like anyone to visit, please go ahead and type that into the chat box, and so people can follow up or continue the conversation. I believe this conversation will be continuing on Discord in the homeroom chat. Yeah, we're all in this together, as Emily mentioned, so we want to make sure that we get this right. I believe that's all I have. I really thank you all for your time and for this great discussion. Thank you, audience, for your amazing questions and the great discussion. So have a great rest of the day. I think we're probably out of time.
Thanks for inviting me. Sorry to be late, and keep planning for a wonderful future in space.
Yes.
All right. Thanks, everyone.
Thank you.